Hey, I think we'll get started. We are the last session that is standing between ourselves and a glass of wine or a nice cocktail. And so I very much appreciate everyone's patience and sticking around with us. It's a pleasure to introduce the next presenting company, VBL Therapeutics. VBL is a company that comes all the way from Israel. Dr. Dror Harats is the company's CEO. He's going to tell us a little bit about the company. It has, I think, a really interesting technology platform. It's a viral mediated technology for disrupting angiogenesis. So Dror's going to talk about that. And the technology is interesting in that it has advanced to the phase 3 stage in glioblastoma multiforme. So Dror, why don't you tell us a little bit about your company?
Thank you very much, Charles. So briefly, I will talk about the company and then we'll have a discussion. So basically, our mission is to develop a game-changing, innovative anti-cancer therapies. And we are already a phase 3 company in recurrent GBM. We are expecting top-line data in 2017 for this billions of dollars market. And we believe that we are potentially the best-in-class going to show the best-in-class data in recurrent GBM, and I will explain why we believe so. We have a novel mechanism of action that I'm going to elaborate a little bit more. We have a very strong IP. Our composition of matter goes all the way to 2033 without any extension. And we are actually working with the top enrolling sites and leading centers in the United States from the beginning. We are an Israeli company, but we are on the NASDAQ here in the United States. Everything that we are doing is under an IND, and most of our trials are done here in the United States. And we believe that there is a great opportunity for value creation. And of course, we have a follow-up programs and a pipeline that's going basically to help the company build up. So what do we have on our plate? We have a platform technology, and from that we go to our lead compound VB111, which is now in phase 3 in recurrent GBM. We have a program in ovarian cancer where we are wrapping up a phase 2 trial. We have a thyroid cancer program with the same products that we already showed that we met the primary endpoint. And we in phase 1 showed that this is actually working in different solid tumors. So we have a gene therapy that's actually working very well in different solid tumors. We have another program that's going to be the next generation VB511, and we have other programs that coming from this platform technologies that actually, instead of destroying blood vessels, enable us to build stable new blood vessels that will work very well in ischemia. Because we are focused on oncology, we are not going to develop it ourselves. We have another program that we didn't disclose yet to the public, which is going to be a very interesting program in immune oncology. How does our vector is working? Actually, we are using an adenovector because the heart of our technology is a semi-artificial promoter that everything that you put behind it is expressed exclusively in angiogenic endothelial cells. So that enables us to use a very strong vector, a viral vector, an adenoviral vector, give it systemically as a simple IV infusion every 2 months, and yet gets expression in a very precise way, specifically in the site and the time that we need it. What we arm this vector with is actually a deadly gene that will destroy these angiogenic endothelial cells. So the way this drug is working is actually the moment that the tumor trying to build its own new blood vessels, and I don't care what type of growth factor the tumor we're using, this gene is going to be expressed and the cells are doomed to die, and we are not enabling the tumor to keep on building blood vessels, and therefore the tumor cannot keep growing. Beside of this, putting this viral infection within the tumor create an immune reaction within the tumors, actually bring up CD8 cells to the tumor and create an immune reaction and actually help the immune system fighting the tumor. So as a company, as a public company, you always try to meet the expectation and actually deliver what we were promising that we are going to do in 2015. And indeed, we did that. We actually showed in a phase 2 in recurrent GBM that we met the primary endpoint. We almost doubled the survival of expected patient in recurrent GBM. We actually promise that we are going to launch the phase 3 trial, the GLOBE trial, the single needed pivotal trial under an SPA and a fast track that we were allowed to start, and we did start it in August last year. We showed that we met the primary endpoint in the phase 2 thyroid cancer. We showed that we got positive results, actually 60% response rate in ovarian cancer, in well-advanced ovarian cancer. We secured the IP for until 2033, and actually we secure our financial position all the way to the first half of 2018, where we are going to be beyond the results of these important clinical trials. We are looking forward for 2016, where we are going to have 2 interactions with the FDA, end of phase 2 meeting in ovarian, and a discussion with the FDA on access program in recurrent GBM. And actually, we are going to present new data in the second quarter, both on recurrent GBM and on ovarian, and later on the thyroid cancer program. But the most important thing is an interim analysis that we are going to show in the first half of 2017 on the GLOBE trial, which is our phase 3 registration trial. And as I explained before, we believe that this technology, beside of being a platform technology, both for oncology and for ischemia, just in oncology, we know that it can work in different solid tumors, and we are now in 3 different programs. In glioblastoma, where we show significant increase in survival. In thyroid cancer, where we showed 6 months progression-free survival, that was a primary endpoint. Later on this year, we are going to show survival data. In ovarian, we show 60% response rate, and we are following the patient for survival, and we believe that there is significant effect of this drug in different solid tumors. So I will end up with this and let Charles ask me some questions.
Well, certainly I can do so, but if anyone in the audience has a question, please raise your hand, get my attention. I'll start though. Dror, first of all, as you know, very intriguing platform, but GBM is about the hardest of the hard. Maybe pancreatics right up there, whatever. And we were recently reminded of that with another company called Celldex, who had a challenge in their GBM trial. And so I guess my question is, how do you view the VB111 program as being different? What is it that gives you confidence to continue to invest in this program?
I think that the major thing in recurrent GBM, which makes it so difficult, is that basically the tumor is almost doubling itself every 2 to 4 weeks. And early in the program, we showed, and that's why the FDA gave us a fast track very early, and the SPA, that we are actually blocking the growth of the tumor. And because this tumor is very unique, it's grown in a closed space in our skull. And because it's growing very fast, it's actually what kills the patient. So if you can stop the growth of this tumor or attenuate it dramatically, you should actually translate it into survival. And because we could show that we actually, right now we know in 83% of the patient with the right regimen, we can block the growth of the tumor or shrink it in a dramatic way. Doing that actually gives us a very good chance to be successful in our phase 3. The second thing that we did is actually we tried not to select at all for any patient in the phase 2. So we allowed recruiting almost dying patient with multiple primary in the brain, patients that had brainstem tumors, patients that couldn't be operated in the beginning, patients that after the chemotherapy and radiotherapy had a recurrent very quickly. And all of this is the opposite of the inclusion criteria that Celldex selected to use. And when you use a very selective population for the phase 2, you cannot compare it to historical control, and then you're doomed to fail in your phase 3. We did the opposite. We took the most severe cases for our phase 2, so we won't spend $20 million on something that wouldn't work.
That makes sense to me, and it's helpful. Let me ask you how the trial's going so far.
So the trial is actually going according to plan. We are doing it in major centers here in the United States. Because the drug is working so well, we got pressure from the Israeli patients, so we have centers in Israel as well, and we are planning to start some centers in Canada in the second or the beginning of the third quarter.
So not a lot of drugs out there for GBM, but one of them, though, most recently approved is a little drug you might have heard of called avastin. So it's intriguing to me that that being a vascular disrupting agent. I guess my question is, how do you see your technology as being different?
You know that avastin was based on a response rate. That's how it was approved. It didn't show increase in survival, and it didn't show increase in progression-free survival in GBM. But because it's such a deadly disease and there is nothing else, it got an early approval, and yet they had to show that they increased survival. They failed to show that they increased survival, not in recurrent GBM. They elected to do the trial in newly diagnosed GBM. Yet the FDA didn't pull the drug from the market because there is nothing else. But when we do a formal comparison to avastin, a full meta-analysis by an independent academic center that's showing that actually the avastin median overall survival is 9 months, and we get to about 14 to 15 months. That's almost doubling survival. I believe that we are going to show survival and being completely different from avastin.
Okay. And with regard to your trial, in phase 3, you say that you haven't restricted enrollment in terms of certain patients, but you have been savvy, in my view, in that you plan to stratify based on age, progression number, other factors that could be confounding variables in GBM. What other factors did you consider? What about primary tumor site and mutational status? Or do you think that your technology supersedes all that?
So because the trial is under an SPA, we had the advantage of a thorough discussion with the FDA on the protocol. And actually, we were thinking what else we can stratify it for. And unfortunately, in GBM, there is not much that is proven that you can stratify it for. So we have a big list of things that we are going to do as exploratory, but we agreed with the FDA that we can only pick up 3. And age is very important, and actually, so that's why we picked these things. But I don't think that we are going to be different if the patient have different mutations, because so far we couldn't find any relationship, because as I said, the tumor need to grow, and we are not enabling the tumor growing. So even if the tumor aggressive and we are blocking its ability, of course it's not going to prolong it as much as we want, but it's very important that we are not selecting actually.
Okay. It's helpful. We only have a few more questions or minutes. Are there any questions from the audience at this point? Yes, George.
You said the technology is independent of the growth factor, and you extend the progression-free and the overall survival. What does the tumor do to overcome your VB111 effect? Do you know?
Why does the patient fail, you ask? Yeah. So actually, that's a great question that we are trying to find out. And you know that in GBM, the cells can grow directly into the brain and actually spread into the brain without creating a lesion. And that, of course, is independent of the vasculature, and eventually it will happen. And the real answer is that we don't know yet. We are not sure why, at a certain point, the patients stop responding. Having said that, I have to say something which is very important. We are doing the protocol, the phase 2 protocol was that first the patient get VB111, and because we wanted to allow them to get enough exposure when they progress, and most of them didn't really progress, it was just edema because we destroyed the vasculature, we allow them to stay on the drug, but they went on the standard of care, which is avastin. So we got a combination of our drug and avastin. When the patient failed, they didn't fail like when they failed with avastin, because when you failed on avastin, there is a cliff. Basically, the patient lived for only a couple of months, maximum 4 months, and the tumor is growing without the flare. In our case, even though it's a combination with avastin, the tumor grows with a normal flare, and the patient lived for almost a year after what we call a failure. And when we didn't notice it, but the doctors who did the trial noticed that the tumor is growing, but growing in a much slower pace than you would expect. And now that we have the spiders of this patient, we do see it. So what we do see is in the beginning, sometimes the tumor is growing, like with immune oncology. Then it stops growing, then it's actually subsiding. At a certain point, it starts growing again. but not in the very aggressive way, in a much slower way, and that's why we get a much longer survival. That's gives me to think that part of it is actually the growing into the brain and not the tissue itself that's growing dramatically.
That's helpful, Dror. And last question, we're just about out of time, but I think you mentioned this, you have sufficient cash to be able to get to a point in which you turn over that card on that phase 3 trial?
Yes, actually we were lucky to do a secondary toward the end of 2015 and raise enough money, so right now we have money to go all the way to the end of the first half of 18, and at that point we will have the full data of this trial and we will be able to go forward in what we need.
Excellent. Thank you very much, and thank you to the audience for your attention today. It's been a great day.
Thank you.
Look forward to seeing you next year.